Coming before you now is Dr. Vasconcelos, yes, who is here today to talk about launching a space biology program at JMU. So I said we were going from international to interstellar. Here we go.
Well, hello, everybody. Thank you so much for having me. And I'm excited to present to you today this project of launching a space biology research program here at JMU. So space travel has been really fascinating and captivating, and I believe uniting humankind since its inception. In the 60s, we were remarkably landing in the moon, developing the technology for all of this to happen. In the 80s, we had all the space shuttle programs that had a wide variety of mission goals that were achieved throughout many years. And these were remarkable achievements where we developed technology, humans went to space, and currently, the current challenge, or where we currently are, is with long-term stays of humans at the International Space Station. So humans have been going and staying for up to around 1 year at the ISS. Now, this is the current state of art. Now, where we are also currently is in starting the Artemis program missions to the moon. The Artemis 1 mission started last year. It was launched and successfully achieved its mission objectives. But it's really just a stepping stone in a series of more complex missions in the next couple of years. The next Artemis mission goal is to have a very similar way around the moon, but with a crewed ship. And eventually, we plan to land again in the moon. Now, once that happens, we'll be experiencing 17% of Earth's gravity, and with the intention of longer and longer stays at this environment. In addition to that, another long-term plan and goal in terms of space travel is really going to Mars. When we do that, we'll have near zero microgravity during cruise and 38% of Earth's gravity when we are on Mars. Now, while this is a very exciting time to be interested in space biology, in space health, and in space flight in general, there are many challenges. The ones that I'm particularly interested in are how the space environment will affect and alter the human body as we grow, as the space missions grow longer, as humans stay for longer-term missions in space. Now, I'm particularly interested in how physiological and psychological effects and how that will change as mission lengths vary in length. So just as an example, to travel to Mars and back would require between 12 and 20 months in microgravity. Now, that's a very wide range of months because it's based on different estimations. It depends on the ship you're using, the rocket you use to launch, when you go, a series of factors. Now, once we get to Mars, we'll be staying either 1 to 3 months or 16 months at 38% of Earth's gravity. Now, ideally, you really want to try to stay the 16 months in order to fully explore the environment and have enough time to perform experiments. Now, how we'll achieve these long-term missions and bring astronauts back home safely is the challenge. Now, what do we know about space health and its effects in human health so far? The space environment affects human health. The two main effects or causes of issues are radiation that can deplete the immune system and potentially cause cancer, and microgravity, which can cause a wide variety of effects. Now, if you just think of astronauts seeing microgravity, there is in addition to being weightless, right, which it looks fun, but there is also a shift of all the fluids from the extremities to the center of the body and to the head. That causes dizziness, balance problems, cardiovascular problems, and also because we're not walking the same way we walk on Earth, there is a lot of muscle atrophy, and what I'm particularly interested, loss of bone, effects in the bone metabolism. So astronauts in microgravity, they lose bone mass during spaceflight and after, at developing a condition that is called disuse osteoporosis. That is similar to the age-related osteoporosis that we develop here on Earth, except it's happening at a much faster pace. Astronauts lose about 1.5% of bone mass per month that they stay in microgravity. And what are we doing as countermeasures, or trying to mitigate some of these issues, in particularly focusing on the muscle atrophy and the bone loss? Currently at the ISS, at the International Space Station, astronauts exercise 1 to 2 hours a day to counteract this muscle loss and bone loss. Except we don't think that will be sufficient long term. The traveling to Mars and longer stays in space will be much longer than what we have ever experienced before. Also, there are two main concerns in these longer-term flights, that the ships will be much smaller, so it will be harder to fit larger equipments. Now think if you're going camping for 2 years and how much supplies you have to bring, right? That's the challenge in terms of mass that we can carry and that we need to transport for a long-term spaceflight mission. In addition, we'll have to have astronauts who are motivated to keep exercising 1 to 2 hours a day for 1 or 2 years. Now, if you just think of how long or how many times throughout the year we don't feel like going to the gym, right? I will not answer this question. Astronauts are humans. They will have to keep that motivation throughout the whole mission and keep exercising at a really long amount of time in order to hopefully counteract the muscle and the bone loss. In reality, we don't think that will be enough, and it is really thought that for longer-term space missions, we need to develop novel countermeasures, which would be, for example, medication or food supplements. Now, we are not only talking about the bone loss. Astronauts also are more susceptible to have bone fractures, and it is thought that these fractures heal much slower when they are in space. So there is a really high risk of an astronaut becoming incapacitated during the mission, and as a consequence, would have a critical risk of mission failure, right? So not only focusing on possible countermeasures and doing this research will hopefully help support space travel, but also we can translate some of these findings to age-related osteoporosis that we develop on Earth. Now, as you can imagine, I'm interested in bone biology, right? So my research program main focus at the JMU Department of Biology is bone biology and investigating human normal bone formation, as well as ectopic bone formation after injury. Now, because of our research focus and our interest on that, I think we are really competitively positioned to investigate bone biology research questions, thinking of microgravity conditions and under simulated microgravity. So this is what we are planning to do as a research approach, is culturing cells in simulated microgravity. Now, in the lab, we use some cells called bone marrow mesenchymal stem cells. These are stem cells that we can differentiate in the lab into the bone lineage. And this differentiation takes around 28 days. So microgravity can be studied in a few different ways. On Earth, we can use fault towers, where we have a few seconds of real microgravity. We can use parabolic flights, where we have a few minutes of real microgravity. But since our cultural system is 28 days long, we need longer-term experiments. So our ideal place to perform experiments would be the International Space Station, where you can have days or months of real microgravity where these cells are exposed to. But you can imagine that getting a payload funded to go to the ISS and perform experiments is competitive and is expensive, right? So we really need to first demonstrate feasibility, build credibility in the field in order to be competitive for these types of experiments. So investigating simulated microgravity would really give us the opportunity to investigate bone differentiation and bone-related effects of microgravity in a way that we cannot do using standard cell culture systems. Now, here's the big picture. The reality of different space environments, right, with different levels of gravity in comparison to Earth. And what we want is to really create and establish a cell culture model with these cells where we are simulating microgravity conditions on Earth. Now, we'll start with a very ISS kind of environment in order to use that as a stepping stone for more intermediate environments. And that's how we plan to do. What we want to do is using these bone marrow mesenchymal stem cells and culture them in simulated microgravity. Now, this is the conventional way that we culture cells. The cells here are in this flask. This is a picture inside the incubator that we have in the lab. They are kept at 37 degrees Celsius, which is the optimal temperature for growth. And the cells are mixed with this pink liquid here, which is the cell culture media. We mix them with the media, put in these flasks, put them in the incubator, and because of gravity, they spread around and attach to the surface of this flask. And this is how they look like when they are attached. This is a similar flask, different type of flask, but the pink liquid is inside here. So our plan, or what we want to do, is to culture these cells using equipment called the clinostat, where you would put a flask similar to that inside this chamber, and this constantly rotates in culture in this direction and this direction. So constantly rotating the cells in culture eliminates the effects of gravity, simulates microgravity, and they cannot attach anymore to the flask, right? We currently use these cells in the lab in bone-related projects using these normal culture conditions in projects that are led by our undergrad and graduate students. Now, I'm a cellular molecular biologist, so I'm interested in gene expression and protein expression. So what we plan to do is, for example, compare the cells cultured in simulated microgravity to the cells cultured in normal culture conditions and investigate which genes are being changed, which proteins are changed. Are genes that are upregulated or downregulated? Is there any effect in the bone differentiation? And from that, select pathways that we can, for example, test drugs that we can intervene, and so on. This is like the stepping stone for future projects. So I believe this project, starting this research program, will have a lot of potential impact. At JMU, we'll be establishing a new space biology research program that blends cell biology, molecular biology, and space biology in a new way that has not been done before. And we will be using that to educate and mentor students in space biology, and I hope opening the doors for new career directions and opportunities. Now, there is a lot of opportunities in the private sector, in the government, or for students to really start their own companies in the field of space biology, space health, that if they are not exposed early enough, they don't even consider as a possibility, as a career opportunity. For the scientific community, I hope that this research will give us a better understanding of bone maintenance and health, and in general for the society, that if we are able to translate these findings, we'll be able to hopefully prevent and/or treat osteoporosis, both age-related osteoporosis that we develop on Earth and disused osteoporosis that astronauts develop in space, and hopefully, in some way, would be helping to enable long-term presence of humans in space. Now, this is my research group here at the Department of Biology at JMU. The group is predominantly comprised of undergraduate students. I have one grad student at the moment, and they are not all, but predominantly biology and biotechnology majors. Now, we'll not be the only ones in Virginia doing space biology research. There are a few other groups, these are the ones that I know, that are doing mostly space health-related research. So there's a group in the Eastern Virginia Medical School investigating the effects of radiation and in sleep, social isolation, and resilience. one group at the Virginia Commonwealth University that is investigating the effects of radiation and microgravity in bone loss and fracture healing, and one group at the UVA School of Medicine investigating the effects of radiation in clonal hematopoiesis and the hematopoietic stem cell. Now, all these groups are using animal models to perform their studies in some way. And I really aim to focus my research program in the effects of microgravity in bone biology, in cell systems, in cell culture systems, which I believe will allow us to really create a niche for us in this research field, and would allow us to develop a novel and unique research program in the state of Virginia. Hopefully, as we establish ourselves, we will also have the opportunity to collaborate with some of these more established groups. So this is my join me to launch this program pitch, okay? My project budget includes resources for the equipment, and this is directly targeted at the clinostat. It's a critical piece of equipment to really simulate microgravity, to culture the cells in simulated microgravity, and to be able to perform all this endeavor. And the remainder of the budget is geared towards supplies and materials, which are really laboratory reagents. So we need laboratory reagents to perform all the cell cultures themselves and perform what we call the molecular biology experiments. So looking at the gene expression analysis, protein expression analysis. Now this budget will allow me to get the program started, optimize cell culture conditions, and start 2 to 3 undergraduate level research programs that would then involve 2 to 4 undergraduate students. Now, this is not a 1-to-1 ratio because some students share research projects, since they depend on how much time they have to dedicate for research in any given semester or year. And if I do have additional funding, it would allow me to expand the number of students that I can get involved in research projects over time, and would also allow me to expand the amount of the types and number of cells that I can use that would allow us to expand the scope of the research project and the types of questions that we are asking. So I would like to thank my research group and the many people who directly or indirectly allowed me to be here today, in particular Madison Trust for selecting me as a finalist and help me prepping for today, and you for your attention. So thank you so much.
Thank you.
Yes.
Where do you get your cells?
Oh, these cells, they are cell lines. They are human bone marrow mesenchymal cells. It's a cell line that has been established, so they are commercially available. So currently, all the cells that we have in the lab, they are commercially available.
Wow.
This slide goes to the point of what I was going to ask. You are partnering with NASA.
I have been trained by NASA. I hope to get funding from NASA at some point in the future.
That was kind of my question. It seems like this is something that a lot of people might be in at this point.
Yes,
And
absolutely. So there are.
Yeah, when you're working with the people within Virginia, so that's good, and NASA, so that kind of answers my question. Okay.
Yes. So yeah, I have been part of a training course that NASA has to train biologists, biomedical translational scientists, really to understand the challenges of developing a spaceflight experiment. It's not what we learned, even what I learned in grad school, or how you plan a normal experiment. Like there is way more that goes into it. So they have a training course to try. They really want to expand the number of people involved doing space research, space health, and space biology. So it was that. Yeah, it's a training program. Yes.
If you run these experiments and you're able to, there's a meaningful measurement in the gene expression and the cellular expression. What do you do with that data afterwards? What does that tell you?
What does it tell me? So the way that I think about it is really like trying to find pathways that we can target for the countermeasures. So if we found a gene or a protein that's, let's say, upregulated, is there any FDA-approved drug that targets that gene or pathway that then I can treat the cells and see if I can mitigate those effects? Can I find something? Can I develop a new drug? Ideally, it's very interesting to repurpose FDA-approved drugs so you don't have to go to all the approval process, and so it's a much shorter timeline. But then finding pathways that we can target, either in the up direction or in the down direction, can you overexpress? So that's how we think about it.
Thank you.
And that could hopefully be a potential countermeasure, stepping stone wise, right?
Quick question from Zoom. Are there limitations to the conclusions that can be drawn from the clinostat versus the animal model, and kind of in reverse? Or would the clinostat be able to provide insights that you can't get from an animal model?
That's an interesting question. I think they're just different systems. In reality, you need complementary information. So with the clinostat, we can really ask in vitro questions. It's much easier to manipulate a cell than it is to manipulate an animal. So you kind of need both, but you can ask very specific research questions, and it's basically a stepping stone. Usually you also don't go to an animal model without having some in vitro data first anyway. So it would be in any way the place to start, in my perspective.
Other questions? Well, thank you very much.
Well, thank you so much. Thank you.